Well, hello and welcome to Inside the Alzheimer's Lab, brought to you by Cure Alzheimer's Fund. I'm David Schenk, author of The Forgetting and senior advisor to Cure Alzheimer's Fund. It's a not-for-profit scientific consortium dedicated to ending Alzheimer's as soon as possible. We bring you this series of discussions throughout the year to help improve the public understanding of Alzheimer's disease. Today we're going to be talking to Dr. Alexandra Newton, who has made some breathtaking discoveries about a protein that has a very interesting relationship to both Alzheimer's and cancer. And we're going to be talking about that surprise connection there a lot and trying to understand what it means. So let's say hello to our special guest today. Dr. Alexandra Newton is professor of pharmacology at University of California, San Diego, where she studies the molecular mechanisms of how cells talk to one another. She received her Ph.D. in chemistry from Stanford University and her postdoctoral training in Daniel Koshland's laboratory at the University of California, Berkeley. Welcome, Dr. Newton. Thank you so much for joining us today.
Thank you. It's a pleasure to be here, and I look forward to the discussion.
So you are an expert, perhaps the world's foremost expert in a protein. Maybe it's not obscure to you and your colleagues, but it's certainly obscure to the general public. It's called protein kinase, or as we write it in shorthand, PKC. And I gather you have actually spent a good part of your career studying this one protein, which, as I understand it, is really kind of an informational protein. It helps cells talk to one another. If you can kind of address just in general what this, for a general audience, what this protein does, and then we'll get how it relates to these various diseases.
Absolutely. So you're right. I have been studying this for my entire career, which is a long time. So perhaps I'm overly specialized, and it does exactly what you say. And so that, when you introduced me, you said that we work on cells, getting information from one another and interpreting that. So this particular protein, protein kinase, or abbreviated PKC, transduces or interprets information within cells. And this whole field of interpreting information and how cells do that is called signal transduction, so the transduction of signals. And I like to use the example of vision, which is not something that we work on, but it's a very easy to understand example of signaling. So right now your eyes are open and photons are going into your photoreceptor cells and you can see. So in order for you to see, all this information has to be processed and your brain's able to read it. Well, protein kinase is one of the proteins that allows this information to get processed. Well, this processing has to be really tightly controlled. When you close your eyes, it's really important that you stop seeing, otherwise things would get very confusing. So signals have to be transmitted very effectively and faithfully, and they have to be terminated very effectively and faithfully. And that's what protein kinase does. It allows signals to go and allows them to stop.
So how many different types of proteins are in this class of protein, these signal transducers? Is it a whole bunch, or is protein kinase?
There are a lot.
Okay.
There are a lot, and specifically, protein kinase itself has 9 members in its family, and they're named by the Greek alphabet, alpha, beta, gamma. So there's 9 protein kinases. But what it is enzyme, so it catalyzes reactions, and it covalently attaches a molecule of phosphate onto other proteins. And when this molecule of phosphate goes onto the other protein, it alters its surface in such a way that it can't bind or interact with things down the line as well, or maybe it'll bind better. It does something different. So enzymes that covalently attach phosphate are called protein kinases. And there are approximately 500 protein kinases.
Wow.
And they're doing the same type of thing. And then there are other classes of proteins that are also transducing information. Probably the main currency in the cell is this moving phosphate around. And these are the kinases So ours is a small part of the big tree.
So I have lots of different questions now. I'm not sure where to go, but one of the things I want to know is, how did you choose to study this particular class of proteins and this particular protein? Was it the connection to cancer that originally led you in here, or were you brought down? Were you brought into this interesting area through another path?
It was through another path. So I had done my Ph.D. In biophysical chemistry, working a lot on membranes, and I decided to do my postdoctoral studies with a very renowned biochemist, Dan Caution, whose name you mentioned earlier on, at UC Berkeley. And he was very interested in solving memory. I had no background at all, obviously, in memory. And at the time, protein kinase C had just been discovered. And it turns out that it's very abundant in the brain. It was originally purified from the brain. And because it covalently modifies and alters other proteins, he thought, wow, maybe this is like a key memory molecule. Let's find out how it works. So I started my postdoctoral studies trying to understand the biochemistry of this new class of kinases, with his goal of solving memory. And we kind of would have this joke in the elevator where he would say to me, "Have you solved memory yet?" And I would go like, "Yes, but I forgot." And so I did solve memory, obviously. I didn't even work on memory in the end. I just understood or examined the biochemistry of how this protein works. And to me, it's a fascinating protein, so we spent over 25 years understanding the nitty-gritty details of what turns it on and what turns it off. So by understanding these nitty-gritty details of what turns it on and off, we are now in a position to understand in diseases like cancer and Alzheimer's and diabetes how this protein is malfunctioning to contribute to this pathogenesis.
Right. So for a while, you thought you were really studying memory through this one type of protein, and then at a certain point, I gather the cancer connection came along, and so you thought you were really studying it. That was really the principal, your principal focus. Is that right? And how did that happen? How did it become clear to you that this was really directly involved in cancer in some way?
Okay. So the whole protein kinase C field has been, to quote one of my graduate students, complicated. It's a very complicated enzyme to understand, and as I mentioned, there are 9 family members. And for probably the last 30 years, people have thought that it is important in driving cancer. So it's an oncogene, and when it gets mutated, it causes cancer. And as a result, there have been inhibitors, so drugs that will stop the activity of protein kinase C, in clinical trials for cancer for 30 years. But they have failed. They have just uniformly failed. So we got to thinking, we work on this enzyme, we don't really work on cancer, but maybe we can use this knowledge that we have of the enzyme to find out why are these clinical trials failing. Maybe they're based on the wrong premise, that it's activated in cancer. So a graduate student in my lab, Karina Antal, with her we decided to look at human cancers, to actually go to the tumors, and now there's a lot of information you can glean from sequencing of whole genomes of all sorts of diseases. And you can go online and find, for whatever protein you're interested in, mutations that have been found in tumors of patients, but that are not present in the non-tumor samples of these patients. So if you do that for protein kinase C, there are over 500 mutations that have been identified in all sorts of different cancers for protein kinase C. So Karina started investigating these, and we thought that these mutations would activate the enzyme so that it was functioning better. And we were very surprised to see that, in fact, the opposite occurs, and they are inactivating, so they are loss of function. So right there, that reversed the paradigm that they're oncogenes driving or causing cancer, and indicated, in fact, that they are suppressing cancer. They're tumor suppressors. And for a cancer cell to grow better, it needs to get rid of protein kinase C because it's acting like the brakes to the survival pathway. So you mutate it so it's not active, and you confer a survival advantage to the cell. So we just found this out very recently. This work just got published this year, that it's actually a tumor suppressor. This is relevant because it explains why these clinical trials have failed. Rather than activating the enzyme, or rather than inhibiting it, as all these trials have been based on, it should have its activity enhanced rather than suppressed.
Okay, so if you're watching this because you want to learn more about Alzheimer's disease, bear with us because we are coming to that point in the story, but I want to make sure that this is all real clear and simple for the many non-scientists in our audience. So to kind of sum up what you just said in very, very plain language, and I think you're being very clear, but just to make it to dumb it down even more, since that's the only thing that I know how to do, you're saying that the cancer community saw this connection. There was definitely a connection between protein kinase C and cancer for many years, and they thought that the more that this gene expressed itself, the more that this protein was expressed in this situation, the more it would help the cancerous tumor grow.
That's right.
Therefore, they've been doing lots of studies and coming up with hopefully drugs that will address that. And what you have discovered very recently, and is now I think being widely accepted in your very focused community, is that the opposite is true. Is that when it comes to cancer, the more that this protein is expressed, it's actually inhibiting cancer. So there's an inverse relationship.
I actually have a slide that kind of illustrates this this balance that we have.
Right.
And so if you kind of think, protein kinase C is really exquisitely controlled, and you have to have just the right amount of activity for things to be fine. And if you tip the balance so that you don't have enough activity or too much activity, you get pathophysiologies. So if we could go to the next slide, what we found for cancer is that the balance is tipped so that if you don't have enough activity, that's when you're getting cancer. And that little model there shows a mouse on the top that's got a tiny little tumor that's got proper normal protein kinase C in this model that we have. And in the bottom one, the red thing indicates a mutated protein kinase C. And we can do these studies with mice, and with a mutation we get a big tumor. We're able to use special genome editing technologies and put that back into the mouse, and the tumor now it doesn't grow or is very tiny. So that's the work that we just published that basically showed that not enough activity is giving us cancer.
Okay, so we'll call that breathtaking discovery number one because it really is just absolutely huge. And I promised the audience that we're about to talk about Alzheimer's, but it's really important that we kind of establish this sequence of events. Can you talk for just a minute about how that information is being received in the cancer research community, whether they're recognizing what you've discovered, and whether or not we're going to see some trials, these trials that are going on for various drugs, change course now that they know that the opposite of what they thought was true is in fact true?
Yes. So when the study came out, it received a lot of attention. I don't know from the perspective of the industry. I haven't talked to anybody there to see if they're going to start trials now where they're using activators, but that's absolutely the next way to go. If you actually look back at the literature over the past 5, 10, 15 years, all of a sudden a lot of studies make sense. So we're getting a lot of investigators that are emailing saying like, hey, this now makes sense why our study was kind of giving us data that didn't make a lot of sense, so we didn't publish it. So there's a lot of literature there that supports this idea. And it's almost a shame that it took so long because of these clinical trials. And so and there was an analysis recently showing that patients that received chemotherapy alone fared better than patients who received chemotherapy and the protein kinase C inhibitors. So these inhibitors were put on to ameliorate the outcome, but in fact it worsened it. So it makes a lot of sense, and I think hopefully we'll be able to see trials now where there's a completely reversed approach where activators are added.
Okay, so we'll be talking probably more about cancer, before the program ends. But now let's talk about the connection to Alzheimer's, because the next point in this really interesting story is where a colleague of yours, Dr. Roberto Malino, who people who are familiar with Cure Alzheimer's Fund will recognize his name. We've talked to him on this program. He's a member of our research consortium, and you work closely with him at the University of California, San Diego. And so he does a lot of work in Alzheimer's disease research, which is not something you've spent a lot of time in. But he made some sort of connection here and brought you into this conversation because he thought that maybe there was something, he saw something that he thought might be very relevant. Is that true? And can you tell us kind of how that happened?
No, that's absolutely right. So as you have been talking about, I have not worked on Alzheimer's before. I work on this one protein, which we became very interested in its role in the disease cancer. And if I could have the next slide. So Roberto Malino is a colleague of mine here, and he had come up with this finding that protein kinase C might be important in Alzheimer's. And very specifically, he found that this peptide, A-beta, that is highly enriched, very high levels in Alzheimer's, that in order for it to cause its effects on synapses, which is basically to weaken them, in order to do that, protein kinase C alpha, and I mentioned there are 9 variants of protein kinase C, so this is alpha, the first letter of the Greek alphabet, easier to remember, that protein kinase C alpha is necessary in order for this A-beta to have its effect on synaptic depression. And he did things like add inhibitors to protein kinase C, A-beta didn't have its effect. Animals that were engineered so they did not have the gene for protein kinase C alpha, again, couldn't have synaptic depression if A-beta wasn't present. You could add protein kinase C back and get this synaptic depression. So it became very clear from his work that protein kinase C alpha is necessary for these A-beta effects. So as part of this, this is kind of an example of collaboration in science. He was the expert on the brain, Alzheimer's. We had the expertise on protein kinase C. And the third player in our story is Dr. Rudy Tanzi at Harvard, who probably has also appeared on this program.
Yes, indeed.
And he's done all this sequencing. He's a geneticist who's done all the sequencing on families that have Alzheimer's disease. And what he tries to do is, with these algorithms that he's developed, find mutations in proteins that are likely to be involved in the pathogenesis of the disease. So these are very rare mutations that in the general population are just basically extremely rare, but they're enriched in families with Alzheimer's. So Roberto Malino went to Rudy Tanzi and said, hey, protein kinase C alpha is important. Can you let me know? Actually, I think he was more broad at that point. Protein kinase C, so that could be any of them, is important in Alzheimer's. Do you have any mutations? So if we could go to the next slide, Rudy Tanzi looked at his databases and found a mutation in protein kinase C alpha that had a higher, it was found in 4 families, and I think he's probably sequenced 400 to 500 families. So 4 of these families, their protein kinase C was mutated at a very specific residue. And on that little family tree there in red are the family members that had Alzheimer's, and 7 out of the 9 affected family members had this one mutation where one residue in the protein was altered to a different residue. And I haven't told you too much about my favorite enzyme, but if I can just hold it up right here for you guys to look at. This is kind of a space filling model right here. So the protein has this little cleft right here where all the chemistry occurs. And this mutation that was identified is right in this little cleft where the phosphorylation and all these chemical reactions are going to occur. And so it seemed right away that it was in a pretty critical location. So when Roberto Malino said, hey, there is this mutation right here, what do you think it does? It was like, wow, it's maybe actually going to be one that is an activating mutation, whereas all the other ones we had found in cancer were inactivating. And if I can say one more thing with my little toys, so this enzyme, as I mentioned before, has to be off and only on when it needs to be on, and you've got this balance where you mustn't have too much activity, not enough activity. So the way that it's normally kept off is it's got all these other little modules that bind on top of it and keep it closed so it can't do the chemistry. And then in response to the right signals, it'll open up like that, and now it can do the chemistry. Well, this mutation appeared to be in a place which would keep it more open. So that's why we thought it might be an activating mutation. It was somewhere where it probably couldn't get so auto-inhibited.
Okay, so I mean, this is just absolutely fascinating and really inspiring. And I'm going to kind of back up and tell, as we did before, I'm going to kind of tell my version of this, and we're going to go over it a couple times because I want people to understand what's happened here, and to kind of take it out of science for just a minute. This is really a story about collaboration, about the amazing things that happen in science when you are open to collaboration, and to be just kind of a bald advertisement for Cure Alzheimer's Fund. This is really what our strength is. This is why we exist, is we have this research consortium, which Rudy Tanzi is the head of. Everyone watching this probably knows that Rudy Tanzi is the chairman of our research consortium, and really all the Cure Alzheimer's Fund is a collection of wonderful scientists who have different specialties and aspects of Alzheimer's disease, who've been brought together by Rudy, and who'd get together several times a year and literally sit in a room and talk about what their research is showing, and they talk about the negative results and the positive results and how they can then add to that story. And these are people that are scattered all over the world, and their labs are scattered all over the world, but they actually physically come together quite often to get in this room and say, all right, so what else do we need to know? What's not working? How do we need to add to this story? And this is just a beautiful case of how Roberto Malino, who was working as a part of this, doing a lot of work for years and years, as a part of this this collaboration with Rudy and the other scientists in the consortium, saw this connection. So what Rudy did is Rudy has been in charge of the Alzheimer's Genome Project, which everyone, again, is watching has probably heard of, which is something we've been doing for years and years, which is looking at families who have more than their share of cases of Alzheimer's, frankly, and trying to discover what genes might be associated with that so that we can understand, well, what are the molecular mechanisms that are especially close to Alzheimer's disease, so we can understand the causes and the progression of the disease. And so Rudy has discovered all these genetic connections, and once you discover a gene, okay, well, that gene does a certain thing. It really is connected to a certain, these specialized proteins which do certain things. So now we know a lot about the disease. And it was Roberto Malino who saw that there was a connection, a genetic connection. Well, he saw there was a protein connection, and he then asked Rudy, hey, do you see some genes that are connected to this protein kinase C? Because if so, it really is going to validate some work that I'm doing in Alzheimer's disease. And Rudy said, yes, actually, we do have some genes, and this is all new. This is all kind of hasn't really been out there before. And once they made that connection, then it was obvious to Roberto that he should go to you to investigate, well, why is this so? Why are we seeing this connection? And is that all, am I getting it right?
That's exactly right. That's exactly right.
Okay.
It was three people with very different expertise that came together for one using expertise in three very different areas. Yeah.
It's really one of the great, if I can just take a moment to make a personal statement, and one of the great privileges of being connected with Cure Alzheimer's Fund. I'm not a scientist, I think people know that, but I actually get to go to these meetings and listen to and watch this in action and see these connections take place. And it's not as though this is the only time that scientists are making interesting connections. This is what science is all about. But I will brag that Cure Alzheimer's Fund does this in a pretty special way, and it's really just on a human level a beautiful thing to see all these people who know really, they're absolutely deeply entrenched in their own special field, and to see these connections being made, and to know that it really is advancing, in this case, not just Alzheimer's disease, but probably also the study of cancer. So that is bragging and it is personal, but I think it's really an important point to make about how, what the kind of human interaction is, behind all these.
Absolutely, yeah. No, I agree with you completely about that. Yeah.
So we've had all these affirmative discoveries. There was the genetic discovery, and then Dr. Malino asked you to look more deeply into it, and you did discover, lo and behold, that there really is this clear connection. In fact, the funny part is, again, to kind of dumb this down, whereas in cancer you recently discovered that the more expression of protein kinase C there is, the less cancer there is.
That's right.
When it comes to Alzheimer's, the reverse is true. That is, the more this protein is appearing, the more it's doing its job, it seems for some reason the more cases of Alzheimer's there is. Is that also true?
That's right. Actually, if we could go to my next slide, it's got my balance there again. And so cancer was a disease where you lost protein IC. So not enough protein IC, you get the survival disease. While degenerative diseases are the opposite of cancer. In cancer, a cell needs to survive and proliferate. Whereas in degenerative diseases, of which Alzheimer's is an example, you have cell death. So intuitively, it kind of makes sense that you would be expecting the opposite. So our question here was, with these mutations that Dr. Tansey had identified, would those be activating? And if we could go to that next slide, I had showed you my big blob, blue blob, that was supposed to represent the part of protein IC that does the chemistry. If we zoom into a very specific part of it, in normal protein IC, you'll see that little red thing, residue. It's an amino acid that packs very nicely into the rest of the protein. So it's a nice packing. In this particular family, or in these 4 families where there were 7 individuals that had this one point mutation, that red residue had been mutated to something a lot smaller. So you'll see the green residue on the right-hand side, and you see it doesn't fill that pocket. There's a space there. And so that was our first clue that that was going to allow that part of the protein to be much more flexible and have a lot more dynamic motion. And I don't know if you want me to go in and tell you the experiments we did to show that.
Sure, yeah, go ahead.
So if we can go to the next slide. Most importantly, it's the 2 graduate students in my lab who worked on that, or Karina Antal, who just got her Ph.D., and Julia Callender, and they're holding the different pieces of protein kinase C. So the next slide shows the tools that we had developed in our lab that allowed us to ask these questions. And we had collaborated about 15 years ago with Dr. Roger Chen in my department, whom some of you may have heard of. He got a Nobel Prize a few years ago for developing ways to use fluorescent proteins to spy inside cells. So we worked with him to develop reporters that would allow us to spy inside cells and look at protein IC activity. And the next slide really just shows a movie where we put this reporter in cells, and maybe you can see it's changing a little bit of color. We activate protein IC, and we can do these pseudo coloring and that tells us whether the enzyme is active or not active. And the graph at the bottom just shows that we're able to quantify these pseudo-colored images. Bottom line is, we have tools to spy inside cells and tell us, in the context of the cell, is protein IC more active, less active, not active at all. So if we use these fluorescent technologies and you look at the graph, the red line is the protein kinase C that's normal. And the green line, which is above it, is this Alzheimer mutant. We were really surprised and interested and happy with this result because it was the first example that we had of an activating mutation. So the green line is on top of the red line, so we have more activity. Everything else we've done so far was the cancer mutations where that mutated line, the green one, would be below, or there wouldn't even be a line at all. We would have complete activity. So very interesting that by putting that little smaller residue there, more flexibility, enzyme gets activated more easily, and we can detect that.
Mm. So, Dr. Newton, what are the tools that you're using? We actually saw one picture of your lab, but we didn't dwell on it. We didn't focus in on what's actually sitting on the desk. When you walk into your lab in the morning, you put on a lab coat, presumably. What are you working with? Are you mentioned that you've got this fluorescence going on, and you mentioned seeing something. So I assume there's some powerful microscopes involved. But what are the kind of basic tools that you're using in order to see these things?
So we basically are a biochemistry lab. So a lot of what we do is biochemistry. And biochemistry is done with pure proteins. So we will take protein kinase C, we will purify it from cells, and probe it, ask questions like, what do we need to do to get enzymatic activity? I told you that what it does is, here's this little Pac-Man model of it. Substrates will bind in here, and phosphate will be incorporated onto the substrate. So that's called a phosphorylation reaction. We can measure that in the lab by using radioactive P32. And so a lot of what we do is we'll take the pure protein, we'll take a substrate, give it P32 labeled ATP, which is basically the currency of the supplier of phosphate in the cell, and just we can measure the radioactivity to tell us how it's modifying these proteins. But we also do a lot of cell biology. So we'll go into cells, and the movie that I just showed you is probably would be more classified as cell biology because we put this fluorescent reporter into the cells and our mutated protein kinase C, and then we can visualize under the microscope changes in fluorescence, which we can then pseudocolor, and we can quantify it, get numbers out of the data, so we can look at the rates and the amplitude of activation. So we do a lot of that. I have another project in the lab on how signals are terminated by something that removes phosphate. For that, we have animal models, so we have mice that are lacking this particular protein. So we go from mice to cells to pure proteins.
And can you give us an idea of how this works in the scope of time? Is it does a project take a week, a month, 6 months to go from beginning to end? Do you have to wait for certain things to happen before you take a look at them? Presumably with mice, you have to read the mice and so forth, but what about when you're not using mice?
Yeah, no, that's a really good question because we always want things to happen faster.
Right.
Some experiments you do, like incorporating P32 phosphate, you can do in an afternoon and you get your data immediately. Mm-hmm. But that's the most simple experiment. Pure protein, pure substrate, mix them together and you get your result. Other experiments with mice can take many, many months to years. So there's this whole spectrum of things. But to give you an example of a project, I received funding from Cure Alzheimer's to work on this particular project last June. And that's pretty much when we started working on it. So between June and now, that was a time that was required for us to have enough data that it's probably ready for publication. A lot of that data also is from Roberto Malino's lab, a lot of it from Rudy Tanzi. So together, that whole group probably with the background from the Malino lab was maybe like a year, and Dr. Tanzi's stuff. So I would say in this case, if we were to add everything together, maybe it's like a 2 years of 3 investigators working on it at different times.
It does start to, for non-scientists, you think about that, and it starts to really paint a picture of, those of us who are on the other side of this are just kind of constantly asking the question, well, when are they going to cure Alzheimer's? And I hear about this big discovery, so when do we get the drug? But then you climb into the details and you realize, well, just getting specific answers to just tiny little pieces of the puzzle, that can take 6 months, that can take a year, and then you've got to ask another question and do another experiment, and it's just
Absolutely. So I'll give you an example. So to get a Ph.D. in my field is probably 5.5 years. So there's one graduate student just working really hard, just very, very dedicated, and just work. And they will probably, after this 5.5 years of work, might have 2 or 3 publications. And so that's pretty much a good correlation for how long it takes in science in my area to publish.
So we're getting lots of great questions on Facebook, and people are going to that question link on our website, which is yorn.com/curealls. So let's talk about the practical consequences of this. First, let's talk about the meaning of it, the practical meaning of it. So one obvious question, I think, and we've had lots of people asking this, is so if too much protein kinase C pushes us in the direction of Alzheimer's and not enough pushes us in the direction of cancer, you were talking about how it's important to have that. The meaning of that is it's important to have just the right amount.
That's right.
Is it possible to have? We've had some people saying, well, wait a minute, I have Alzheimer's and cancer in my family. Does that make sense? Does that really comport with what you're seeing? So what would you say to that question?
So that's definitely possible. So as you've just mentioned again, I keep talking about this balance where you need to have just the right amount of protein kinase C, not enough cancer, too much degenerative diseases such as Alzheimer's. So the question is then, can you have both at the same time? So there have been some recent analyses where this exact question has been posed. What's the link between cancer and Alzheimer's? And I guess it's been known for quite a few years, more anecdotally, that there's an inverse correlation. Well, meta-analysis just came out this year where studies from many, many different groups had been put together. And the correlation there was that if you have Alzheimer's, there's a 45% reduced risk of getting cancer. And conversely, if you have cancer, there's also about a 40 to 45% reduced risk of having Alzheimer's. So although you can have both, and that's bad luck, and basically cancer's bad luck and Alzheimer's is bad luck, you can have the two together. The probability of having the two together is lowered. If you've had one, there's a lower probability of having the other. So it's not black or white. You just have your chance. And
just to talk about it with respect to protein kinase C, how would you explain that? Because it sounds like I'm a very black and white thinker, and it sounds like, well, if you have, if you even showed that balance there. So if I have too much, and that means that I'm more likely to get Alzheimer's, how would I get cancer? It sounds like it's going. It sounds like you can't have both. So how would you make sense of that?
So the following way. So cancer is a disease where many, many, many things go wrong.
Okay.
So it's a cancer cell that just basically scrambles to find ways that it can survive.
Right.
So quite often the machinery that repairs DNA goes wrong in cancer. So in a cancer cell, it's not that you just have bad protein kinase C. You have bad many, many, many things. So there might be 100 different things that have gone wrong. And these are mutations that occur during the progression of the cancer, or they're They're mutations that have occurred after the person has been born and lived. So they'll be localized to the tumor. If you have breast cancer, you'll have mutations that are present in the tumor in the breast, but they won't be present in healthy tissue in the breast. So these are cells inside that tumor that have gone crazy. But in a disease like Alzheimer's, these are hereditary mutations, where you received from one of your parents a mutated copy, let's say, of protein kinase C, but probably everything else is fine. Mm-hmm. So if you have this mutated copy of protein kinase C, it's bad for Alzheimer's because you have too much activity and it's kind of favoring Alzheimer's. But that person might still get cancer because they might have a tumor where like 50 other things got mutated. And even though they have this good protein kinase C that's trying really hard to protect that tumor, there's all these other things that went wrong that override the fact that you have this protective protein kinase C.
Right.
So your probability of getting it are lower, but these other things have gone wrong.
That makes a lot of sense. By the same token, do we need to worry once we start putting your knowledge into action and we come up with a drug that says, okay, you don't have enough expression of protein kinase C, so that means you're at a higher risk for Alzheimer's, so let's improve that. Let's give you a drug that kind of boosts that activity. Does that mean we're going to be pushing people then more in the direction of cancer if we're trying to lower the risk of Alzheimer's? Is that going to be a danger?
So another great question, and I can address that two ways. First, as I mentioned, there are 9 different variants of protein kinase C. We know that it's protein kinase C alpha that's the Alzheimer's protein. All the other ones are involved in cancer. You lose any one of those, and that's bad for cancer. If we can specifically target only protein kinase C alpha, all of a sudden you've reduced your efforts on the cancer problem is lowered because the other 8 are there. The other thing is that this particular protein kinase C alpha is bound to a very specific protein. So if we can now narrow down in molecules that are going to inhibit it only when it's bound to this particular, let's think of it as a parking lot in the cell, if we can direct it only to that pool of protein kinase C alpha, again, we're doing very well. And then lastly, if we can get these inhibitors to only be in the brain and not systemically, and not have chemotherapy going systemically, that would also reduce that. So I think there are ways to get around this issue, and they're just going to involve a better understanding of where it's too active and how we can target it at that particular location.
So there's going to be an awful lot of activity now in trying to take this knowledge and come up with a drug. And I gather there are drugs, as you said, that have kind of been going the wrong way for cancer research. But it sounds like maybe that there are a couple of drugs out there that we thought they were going to be useful for cancer, but maybe they will be useful for Alzheimer's?
Absolutely.
Okay.
If we can go to my next slide, and actually I'll just say one thing on the next slide. It turns out that Rudy Tanzi found 3 mutations in protein kinase C alpha. I talked to you about one of them. All 3 of them were activated. So the algorithms chosen to predict what might be important, this is a nice example where 3 out of 3 were all activating mutations. But the next slide is what I really want to show. And that's a cartoon of some people at a drug trial, and the guy standing there is basically saying that our trials show that the new drug performs no better than placebo, and this other guy goes, well, maybe we should invest in placebos. In fact, I told you that placebos would have been better. So what about repurposing these drugs for Alzheimer's, where we tip the balance in the other direction? There is an inhibitor of protein kinase C that failed for cancer that's now in phase 2 clinical trials for Alzheimer's. It's called bryostatin, and it worked very nicely in mouse models, and now we'll see how it works in these phase 2 clinical trials for human patients. But based on what we found, it should be a very effective way for targeting protein IC. It'll cause the loss of protein IC. It's not specific for this variant of protein IC, so we have this cancer issue to worry about, but it's certainly a beginning.
Is there anything we can do in our daily lives, foods we should eat or not eat, exercise we should partake in, sleep, anything at all, meditation, that affects our levels of protein kinase C as far as you understand?
There really haven't been really strong, I mean, basically the whole thing in the cancer and I guess any kind of disease is just to lead a healthy lifestyle.
Right.
And eat your fruits and vegetables and do your exercise and get your sleep. But there isn't something very, I mean, there are studies that antioxidants affect protein IC and all sorts of things. But I think basically we just have to have healthy lifestyles, and even those of us who have healthy lifestyles sometimes will have the bad luck of cancer. So,
right.
Yeah.
Well, but just to reinforce that, I mean, are there, so it's not like you have a study saying if you eat more Brussels sprouts, you'll have, you know, a better protein kinase profile, but do we have studies on a more broad level that say if you do all the things that doctors have been telling us to do, you know, for years and years, you don't have too much sugar and you know, exercise and you eat right, do those people tend to have, I mean, or is this even knowable? Do they tend to have better protein kinase?
No, I think in terms of like looking at protein kinase as a function of lifestyle, that really has not been looked at at all. Yeah.
Should it be? Is that something we can do?
Well, I think if I can go back to quote my graduate student Karina Antal, protein kinase is complicated. So I think there are so many things that can affect it. It would be very difficult to say, well, this person had these mutations in protein IC in their tumors because they did or didn't do whatever. I think it's just there's just too many inputs into controlling protein IC and too many things that could go wrong for us to be able to figure that out.
Here's a great question that came over the Yorn question system. And that is, are there any other diseases that might be connected to this? I mean, it sounds like protein kinase C is pretty central in there. Are there other diseases you know about, or should we be looking for other diseases that are being affected by too much or too little, or the way that it's being expressed?
Yeah, so that's another great question. So if we think back about that balance again, where not enough are these survival diseases, of which cancer is the great example. But then if you have too much, it's going to promote these degenerative diseases. Alzheimer's is a degenerative disease. Heart disease is another great example of a degenerative disease. And protein kinase C activity, high activity, has been linked to heart disease. So yes, we can generalize to these degenerative diseases, which will be promoted by having too much protein kinase C.
We had a question about, this is a very specific question, and if you can't answer it, we'll do that on our website. But do you happen to know where people can get more information about the Bryostatin trial, which is one of the drugs that we've been talking about?
Yes, so there's a company called Neurotrope. And if this person Googles Neurotrope, or even Googles Bryostatin clinical trials, that information should be there. Okay.
If you personally were dealing with Alzheimer's or cancer, would you personally want to follow up on this Bryostatin trial? Would you, try to get in on it, or somehow, at least learn more about it?
I think personally I would be a little hesitant to get in on the Bryostatin trial because of what we talked about, how that will basically cause the loss of all the protein kinases in your body. And so I would be much more interested in a clinical trial targeted at protein kinase C alpha. And I certainly hope that that will be in the works soon. So I would wait for that next one where we can be a little more specific.
Great. So we just have a few more minutes. The audience should feel free to continue to ask questions, and we'll try to get to as many as we can. What are the next steps here? Where are you taking this next? I take it you're not going to go away from us, that there's a lot more work to be done here. But not just, I'd love for you to kind of incorporate in your answer not just what you're going to be doing, but what else do we need to do in the Alzheimer's community to take advantage of the discoveries you and Dr. Malinow have made?
Right. So the next thing that we're doing is we're developing animal models that will allow us to find out if introducing these mutations in protein kinase C are sufficient to cause the Alzheimer's phenotype. The other thing we're very interested in doing is looking at the levels of protein kinase C in autopsied brains of patients who had Alzheimer's. And the prediction would be that they will have elevated protein kinase C, or we have ways to detect if there was elevated activity there. And the reason for looking at that is, okay, so some of these patients who've developed Alzheimer's, they have this mutation in protein kinase C alpha, but other patients don't. Are there other ways that protein kinase C has been activated in these other patients? So is this like a very general thing that if you have Alzheimer's, your protein kinase C is amplified? So we'd like to answer that question. We'd like to also use our animal models to be able to test eventually inhibitors that will be very selective for protein kinase C alpha, this particular variant. So basically understanding how general this phenomenon is, finding from animal models if these mutations are sufficient to cause Alzheimer's, and then using animal models to develop drugs, which hopefully eventually will be able to go into clinical trials.
Well, this is just fascinating. I want to thank you on behalf of the general public for just all the work you've done. It really sounds like you're the right person for the right time, and it's amazing how obviously many years of hard work have now, not that you haven't made important contributions before, but really it seems to be coming to a head now, and it's a great lesson for maybe young scientists out there that that if you kind of keep your nose to the grindstone, that there just could be amazing payoffs in your work in ways that you can't possibly predict. So I think it's a terrific story, and we're lucky to have you, and I appreciate you taking the time to talk to us today. Is there anything you want to add before I say goodbye to folks? There's something.
No,
Okay.
Yeah, it's been a pleasure talking to you, and I hope this makes sense to a lot of people. And I think what you just said, that our lab was basically at the right place at the right time, talking to Dr. Malinow on this and Dr. Tansey, and all coming together to make some very interesting findings.
I should ask you a question. I know that I was in the goodbye mode, but I should ask you a question about research funding. You talked about we've been able to get you some money from Cure Alzheimer's Fund, which is privately funded through donations, charitable donations. But I gather I hope that your lab has also gotten some public funding, gotten some federal NIH research dollars. Is that true? And how does that work?
Yeah, so I have funding from I have federal funding from the NIH, and that supports the research in my lab on the cancer and the basic mechanisms. And the funding for the Alzheimer project has come exclusively from the Cure Alzheimer's Fund. They basically provided seed money to get us in here. And that's been very helpful, and probably without that we wouldn't have had the funds to start this project. So yeah. So two lessons there, I would say, for people watching, listening to this, is number one, we really need federal research. It's the gorilla in the room. There's so much that we can do on a big scale by funding all these basic research projects, and I think people should support public funding of science. I won't apologize for making that statement. And also, we can see that there's room for these very innovative, small nonprofits like Cure Alzheimer's Fund, and there are many others out there, where they're kind of out ahead of the NIH. They're really doing very risky research on a small targeted level, and they're taking chances, and they're exploring things in a way that sometimes a big bureaucracy can't. So it's really, again, in this one lab, we're really seeing such a great illustration of how science can work very well in this here in the capacity of different kinds of funding.
So that's all the time we have for our program today. Thank you so much for watching. If you have any questions about the science in today's show, please do post them on our Facebook page, and we'll try to answer some more questions there. Inside the Alzheimer's Lab is a production of Cure Alzheimer's Fund, a not-for-profit dedicated to supporting the research that will stop this terrible disease. Every dollar you donate to Cure Alzheimer's Fund goes directly to scientific research. That's because our board of directors covers all administrative costs. So to repeat that in another way, if you look at the behind the curtain of nonprofits, usually you donate $1 to their cause, and a certain percentage of that gets spent on their office costs and their staff costs and all that. With respect to Cure Alzheimer's Fund, when you donate $1, that's $1 that will go to a lab like Dr. Newton's or Dr. Malino's or Dr. Tanzi's. The entire dollar goes. That's because our board of directors covers all of our other costs, which is really a fantastic thing. And now we will thank them in person. So thank you to our board of directors. That's Chairman Jeff Morby, Jackie Morby, Henry McCants, Phyllis Rappaport, Robert Greenhill, Matthew Zulik, Sherry Sharp, and our CEO, Tim Armour. Thanks to Dr. Rudy Tanzi, chair of our research consortium. And thanks again to our wonderful guest, Dr. Alexandra Newton, not only for all of her work, but for taking some of her valuable time to talk to us about her research today. I'm David Shank. Thank you so much for watching, and we'll see you again soon.